Hi all, my name is Malia Santos, and first I want to thank you all for tuning in to my talk today. I'm going to be telling you about some of my preliminary results on my work in investigating species relationships and evolutionary patterns of defense strategies in Trichlicia. So to broadly begin, one of the main questions asked in evolutionary biology is understanding what processes have led to the patterns of species diversity that we observe today. These processes, whether abiotic or biotic, all play a role in shaping species diversity. I'm only showing you just a few examples of some variables, but would like to highlight one factor that I'm most interested in and what has really driven me to work in Rubiaceae, and that's herbivory. Rubiaceae, being the fourth largest plant family, is mainly a tropical family. The tropics are a known biodiversity hotspot where herbivores are a source of selective pressure due to the increase in frequency and intensity of ecological interactions. Rubiaceae also possesses many interesting defensive traits, including secondary metabolites such as caffeine, domatia that can be found on the leaves or on the caudex. This can be shown in the left-hand photo in our middle photo. And domatia provide basically a place to live for predaceous or fungivorous arthropods and basically act as a defense system for these plants. And along with this, Rubiaceae also has many instances of foliar bacterial nodules, which serve a similar purpose to the domatia, just with bacteria. And so even though the tropics provide an interesting place to study the effects of herbivory on plant species diversity, the tropics, and especially tropical Africa, where my plants reside, is a difficult place to study due to internal and external conflicts. So here I'm showing you a time lapse of botanical collections from 1780 to 2015 by Sosaf et al. To illustrate that researchers have collected in a majority of places, however, scattered and not really continuous over time. You can see this in the older yellow to orange points that cover a broad geographic area, and red points collected most recently are in these concentrated pockets. And so an issue that arises when using those older herbarium samples, especially in tropical places, is that a lot of plants were preserved in ethanol, which basically destroys any chances of retrieving viable DNA. And so we're kind of left in this spot, well, what can we do? And so in the phylogenomic era, it's become much more important to have high quality DNA. Than when we were retrieving just a few plastid loci with PCR. And many studies have relied on these high copy DNA regions like the chloroplast, which poses problems when looking into resolving species relationships in really complex groups. However, as these sequencing efforts have improved, so has our capability to retrieve nuclear DNA from fragmented DNA, which is often the case when you have plants that are not dry in silica gel. And so this has really resulted in the field of museumnomics, where we can use herbarium specimen, or we can try our hardest to use them. And I say that because it is worth noting here that you're gonna end up with some specimen not working well due to the collecting methods used in the past, like ethanol. So now that you have an understanding of why I want to understand this group more, I'm gonna give you some background on Trichlicia. Trichlicia is a genus of tropical woody shrubs or trees that are located throughout sub-Saharan Africa. They are very architecturally similar to coffee, so much so that you probably didn't realize that the middle photo is actually coffee. It's been said that they're very hard to distinguish or tell apart in the field, but distinguishable by these long needle-like awns at the top of each stipule. And so I have a picture here on the left, but if that's too hard to tell, I have an illustration here on the right for you. So most of the taxonomic work done in the group was done by our co-author, Elmar Robrick, who published a series of monographs hypothesizing relationships between species based on morphology and geography. And in the next slides, I'm going to show you each section and show you some of those morphological characters that distinguish the groups. We'll start off with section Triclasia. As I go through these slides, take a look at the left-hand side where I'll be showing you the species distributions for each of the sections. Section Triclasia is comprised of about 40 species and is considered the core section where many of the relationships between species are pretty difficult to recognize. They're mainly residing in the Guinean-Congolian region, which is hypothesized to be the center of diversity for Triclasia, but are also found on the east side of the continent. To kind of solve this problem of a lot of it kind of just being like a dump for a lot of the Triclasia species, Robrick also split this section up into multiple groups based on some other characters. Section Problytostemen is considered to be a morphological transition between Triclasia based on a number of morphological traits that are not shared with other members of the group, such as free bracteoles, as opposed to being fused into calculi, which is, as opposed to being fused into calculi. And then they also have these large sclerified fruits, which is which differs from those really small fleshy fruits that we see in the rest of the group. Section Ephedranthera can be characterized by sessile anthers. Section Rosea is morphologically very similar to section Triclasia, but has a very narrow distribution along the Indian coastal belt. And finally, section Androgyne, the Malagasy section, that can be characterized by unisexual flowers that appear hermaphroditic, but are functionally male or female. Previous phylogenetic work is scarce and is limited to a study done in 2009 by Tosh et al. That investigated the monophyly of Triclasia using 4 plastid regions. They found that Triclasia was paraphyletic and decided to resurrect Empagona as its own genus to solve that issue. But however, despite the chloroplast data out there, it doesn't provide resolution at the species level. And so to answer, or before we begin to answer this question in this group, we need a robust species level phylogeny. And we propose to do this by adding a nuclear data set to obtain this robust phylogeny. So I'm going to go over my methods very briefly. We decided to use the Angiosperm 353 data set that putatively targets 353 nuclear single copy loci. This approach also allows you to recover complete high copy elements that are appropriate for phylogenetic analysis. The Angiosperm 353 has been shown to generate phylogenetic resolution across various taxonomic levels, and that's why we decided to use this target capture method. We used Tide Piper to process our reads, and we modified our target file using McLay et al. 2020 or the Mega 353 dataset, along with adding Triclasia samples of our own to improve our capture efficiency. Before I continue, I'm going to jump to some of my results quickly. This is a taxon by gene matrix that Tide Piper provides you. And I'm only showing you this to point out that we had some samples that worked pretty well, and we had some samples that didn't work too well. And so we decided to exclude the sequences with low capture efficiency because they just didn't have enough data. We also decided to exclude paralogs for the time being. After doing that, we aligned and cleaned our sequences using MAFT and futility. And we have also tried various phylogenetic methods, but I'll just be showing you the results from my maximum likelihood run in IQ tree. So right now I'm showing you our latest tree overlaid with the morphological and biogeographical sections proposed by Robrick. I would like to highlight a few interesting things. First being Tricolosia comes out as monophyletic with high support. Empagona also comes out as sister to all of Tricolosia, which was found in Tasha et al. 2009. Section Probledostemen was proposed as a transitionary section due to some traits that lack fusion. And so we can see here that it's probably an early diverging clade and maybe sister to the group, confirming Robrick's hypothesis. We can see that section Ephedranthera is scattered, suggesting the evolution of a sessile anther evolved multiple times. We can also see that section Androgyne forms a monophyletic group, suggesting one introduction to the Malagasy region. This is important because in other groups, they have seen multiple introductions into the Malagasy region. And so maybe that will change when we add more specimen, but for now, that is what we see. And then while the morphological sections are not monophyletic and the characters are a little bit more labile than previously thought, the geography of this species shows really strong phylogenetic signal. So the photo on the right is taken from Linder et al. 2014 on the evolution of plant diversity and is showing you a broad regionalization of Africa. We can see that the origin of Tricolosia is centered in the Guinean-Congolian region, which is suggested by Robrick. We also see one introduction into the Malagasy region and then potentially one introduction into the Zambesian region or the East South African. We have some branches that are not supported well, and so some of the next steps for us are to investigate paralogy. We would like to try to use Phylucci because some people have had more success using Phylucci to investigate paralogy. We would also like to filter our data set more. And so I'm just gonna plug one tool that I've been pretty excited for. Joseph Rusinko gave this talk at Evolution and is giving it again at Botany. So check it out. It's at Friday at 3:30 in the phylogenomics section. And this is a tool or a coalescent-based tool for taxon selection in phylogenomics. And so this is what we've been struggling with due to the samples that had pretty low capture efficiency. And so we kind of just excluded those, but we would like to have a more kind of coherent way of choosing samples. Along with filtering our data, we would also like to increase the samples that we have, one in the number of species and then in the number of taxa per species. And then finally, I would like to understand how plant defenses have evolved through this group. And this is my main overarching goal. And with that, I would like to acknowledge all of the botanical gardens that have sent me samples and silica gel, and especially Petra de Bloch at the Mies Botanic Garden and Tariq at the Missouri Botanical Garden, who worked basically through the COVID-19 to get me my samples. Also University of Idaho and iBest. And then just a little plug before I end, the Tank Lab is moving to the University of Wyoming. So if you are interested in joining the Tank Lab, please get in touch with David Tank at the following below. And then if you have any questions, please don't hesitate to contact me. Thank you.